I'm the chair of the Genetics and Epigenetics graduate group in CAMB, and I want to describe this program to you so you have a better understanding of what we're about. Get my slides up here. So the research topics that are covered in the Genetics and Epigenetics program are probably the most diverse of any sub-program. In CAMB, we have people working on the inner workings of the nucleus to identifying human disease genes, as well as those trying to figure out how those or defects in those genes lead to the human diseases that they're associated with. There are three major subgroups in the program. We have human genetics, people who are human geneticists, people who use model organism genetics to study biological processes and diseases, as well as those who study epigenetics and gene regulation, again, to understand basic biological processes or disease state. The leadership of the program consists of a chair, that's myself, co-chair Joe Zhao, who's going to take over leadership sometime later this year, as well as a group of hand-picked advisors. These are some of the advisors we have who know the program well, know the courses well, and will help you in your choice of thesis labs, which is one of the most important decisions you will make if you start graduate school, and also will help you choose the courses that are a best fit to help you with your research. They also will help you guide through many issues that are associated with carrying out your research studies and doing your thesis work. In the Genetics and Epigenetics program, we have about 80 different labs and currently about 65 students in this program. Now, in the next few slides, I want to describe the research of some investigators in this program so you get a feel for the diversity of the research that's performed in our program. So from the genetic basis of human traits and disease, human genetics studies, I'm highlighting the work of Ben Voigt and Sarah Tishkoff. Ben is a computational biologist by trade. He uses statistics and population genetics to identify the genetic pathways that lead to human disease and human phenotypes. Sarah Tishkoff is a well-known human geneticist who has her lab travel to different parts of Africa to obtain DNA from various historically isolated populations to help with studies of human genetics, complex disease, human evolution, etc. And she has become recently the head of the Penn Center for Global Genomics and Health Equity here at Penn. We're very happy she took over that leadership role. Under the heading of model organisms genetics, I'm highlighting the work of Marisa Bartolome and Klaus Kessner. Marisa, as a graduate student, discovered genomic imprinting, and she continues to study genomic imprinting and X chromosome inactivation in her lab currently to this day. And Klaus Kessner uses the mouse to study pancreatic and liver development and their association to metabolism, diabetes. Under the heading of epigenetics and gene regulation, Shelley Berger and Jennifer Philip Cremens are two eminent scientists in this area. Shelley Berger is a pioneer in the field of epigenetics. She's actually the head of the Penn Epigenetics Institute now, and her lab studies how the language of chromatin modifications are involved in basic biology and human disease. She has a wide range of research areas that her lab is focusing on. Jennifer Philip Cremens uses 3D analysis of the nucleus to look at how the genome is organized and how that organization impacts gene expression and how it changes during the specification of a cell or how it's affected in disease state. Under the heading of RNA biology, we have a very strong RNA biology group, highlighting the work of Brian Gregory and Kristen Lynch. Brian Gregory is in the biology department, and he studies RNA metabolism. He has developed many ways to look at protein-RNA interaction and then how this RNA metabolism is impacted in its role of plant function. Kristen Lynch is a co-chair of the biochemistry department and biophysics here at Penn. She and her lab studies how her lab uses experimental and computational approaches to study alternative splicing regulation and its impact on cellular function. Now, this slide here I'm using to highlight one of the many collaborations that exist at Penn. Penn has a very collaborative environment. And in this slide here, it's a child with Cornelia de Lange syndrome, which is a disease that is heavily studied at the Children's Hospital of Philadelphia. Ian Krantz is the world's expert on studying Cornelia de Lange. And Eric Joyce is a faculty member of the genetics department. And together they're studying how the disease genes that cause Cornelia de Lange, which are proteins involved in chromosome organization, are, when they're lost, how they impact chromatin organization. Many years ago, a Drosophila model of one of the human diseases, or one of the genetic causes of Cornelia de Lange was made, and many phenotypes, including dysregulation of circadian behavior, shown down in this panel here, was shown by my lab and Amita Segal's lab. And Eric has gone on to use this model to look at the chromatin organization in this fly model, and using a methodology called oligopaints, he can light up all the chromosomes in the Drosophila nucleus. And here you can see a nice wild type nucleus with the different chromosome arms labeled in different colors with these paints. And if you look at this same nucleus, not the same nucleus, but an analogous nucleus in this Cornelia de Lange model, you see that the chromatin organization is highly affected, and this has pronounced effects on gene expression. And Eric is currently working with Ian to try and understand how the loss of the chromosome organization is affected in this model and can lead to disease state. Now, as students in the program, you will have certain courses that you need to take, but our requirements are relatively low compared to the other programs. In addition to the CAMB required courses, our students have to take a genetics principle course, which covers model organism genetics and human genetics, as well as a course that's an introduction to bioinformatics. Beyond that, there are 2 additional elective requirements, and there's a whole list of elective courses that students can take. Some of the examples and popular courses are modeling human diseases and genetic systems, topics in human genetics and disease, experimental genome science, and a whole host of GCB courses, as well as students are now starting to take computer science courses to help learn how to program for their research needs. Our program has very close ties to the Genomics and Computational Biology graduate group. In fact, many of the faculty in GCB are also in G&E. 35 out of 80 of the 80 faculty in G&E are also in GCB. Because of this close relationship, GCB courses are open to CAMB students, and a lot of the G&E students take advantage of this. And there's also dual mentorship options. Oftentimes now students will find that they have a wet bench side to their research as well as a dry lab side. And to help guide them through their thesis work, they often end up pairing a mentor that's a wet bench mentor as well as a dry lab mentor, and can help. This dual mentorship can help them work through their research most effectively. Since 2011, the G&E students have joined over 65 different labs in our program. We currently have 8 trainees on an NIH genetic training grant that we run with our program. And greater than 50% of our students in the program are successful in obtaining an NIH F32 fellowship to help pay for their research in their thesis work. And upon graduating and leaving, the average G&E student publishes on average 2.76 papers each. That's a very good number of publications. In addition to all of the classes and research that's carried out, there's also a number of activities that our program sponsors. On Fridays, we have a research in progress talk where students and postdocs give 30-minute presentations of their ongoing research to the genetics community at Penn. We have student-sponsored seminars where the students in the G&E program get to invite seminar speakers that they'd like to hear from and get to sponsor that speaker during the day and host them, as well as introduce them during their seminar presentation. We have genetics career forums with alumni, so we bring back the alum of the G&E program who have gone out and obtained jobs in many different areas, and we have them come back and talk to the current G&E students about their career paths and why they did what they did and what they would do differently. We also have discussions in genetics once a month, which covers a wide range of topics, but is a forum where we have informal discussions of various issues that are arising in the genetics field, as well as discussions about how to approach a problem differently based on different people's points of view. We also have a fantastic Final Friday, which is a happy hour at the end of each month. That is sponsored by G&E, and it's a great social activity for G&E students and some faculty to get together and discuss in a relaxed environment. With that, I'd like to thank you for your attention, and if you have any questions, do not hesitate to email me. You can find me on the G&E webpage in the CAMB directory. Thank you.